The overall goal of this procedure is to reliably elicit offensive aggressive behavior in male rats in a laboratory setting. The first step is to house an adult male rat together with a sterilized female for a period of at least 1 week. On the day of testing, video recording equipment is set up to record social interaction. Next, the companion female is removed from the cage. When ready, an unfamiliar male rat is introduced into the cage of the resident male, and they are allowed to interact for 10 minutes. Ultimately, the procedure is aimed at the establishment and maintenance of a territory by the resident male that he is willing to defend against intruders.
The strength of this method is that it allows the analysis of natural social behavior in a laboratory setting, not only in the resident male but also in the intruder.
This method can help answering key questions about the neurobiology and neuropharmacology of biological functional aggression, as well as violence as the pathology of aggression.
The technique can be used to study the consequences of acute or chronic social stress in the intruder.
Please notice that the resident-intruder paradigm is not free from ethical concerns. Serious suffering may occur in the intruder male. Potential animal suffering should always be in balance with the main scientific aims of the experiment. Offensive aggression is a highly active behavior, and therefore sufficient space is required for its full expression. For optimal results, use resident cages that have at least a half a square meter of floor space. To facilitate developing territoriality and reduce social isolation, house each resident with a female for at least 1 week before the start of the experiment.